In Chapter 10, we're going to discuss how photoautotropic organisms can use sunlight energy to generate glucose. In the process of photosynthesis, photoautotropes are going to use carbon dioxide and water to generate glucose and oxygen molecules. Since this is building molecules like glucose, that means it is endergonic and we're going to need an energy source. And that energy source is light. We have a lot of different organisms on this planet that can carry out photosynthesis. Here we can see cyanobacteria, which is one of the earliest cells that we know of that could do photosynthesis. And these are actually really important evolutionarily and historically because they were so good at photosynthesis that they contributed so much oxygen to the atmosphere that it ended up causing a mass extinction event called the Great Oxygen Catastrophe. We also have protists like this dinoflagellate and euglena, which have chloroplasts within them so that they can also carry out photosynthesis. And then, finally, we have some animals that have symbiotic relationships with different photosynthetic organisms so that they can get that glucose directly. For example, the aphid and the salamander. The sea slug at the bottom is really interesting because it appears to have adopted the genes from photosynthetic organisms and incorporated it into its own genome. So they actually do carry out photosynthesis within their own cells. However, your book is going to focus on how plants carry out photosynthesis. Keep in mind that not all plant cells will be able to do this. For example, roots and bark cannot carry out photosynthesis, but the things that are green, like leaves, can. Here we are looking at a cross-section of a leaf. On the upper surface we have a layer of cells called the upper epidermis, and these epidermal cells are producing a waxy substance called a cuticle, which will protect the plant from dehydration, from pathogens like bacteria and fungus, and also from UV radiation from the sun. Just beneath those epidermal cells, we have the mesophyll cells. These are the photosynthetic cells. On the lower surface of our leaf, we have the lower epidermis, and embedded within this lower epidermis, we have specialized cells called guard cells. The guard cells are going to create and maintain the stomatal pores. The stomata is where we have gas exchange between the environment and the inside of the leaf. This is how the leaf would breathe. CO2 enters into the leaf organ, and oxygen, gas, and water molecules could exit through the stomatal pore. The guard cells will swell up and shrink down in order to control how large or small that stomatal pore is. Within the mesophyll cells, we have a lot of chloroplasts, like you saw on the Alodia leaves in the lab. The chloroplasts have two membranes. They have an outer membrane and they have an inner membrane. Inside the inner membrane, there is a fluid called the stroma, where we have ribosomes, RNA, and circular chromosomes as well. We also have circular discs called thylakoids within our chloroplasts. And we have many of them, and they are all stacked up like pancakes, like you see here, in structures called grana. The chloroplast is where all of this photosynthetic activity is going to occur. And as you can see from this diagram, it's a little bit more complicated than simply taking CO2 and water and creating glucose and oxygen molecules. It turns out that there are two big stages to photosynthesis. We have the light-dependent reactions that happen along the thylakoid membranes, and they're going to be producing ATP and a molecule called NADPH, which can then be used in the second part of photosynthesis, which is the Calvin cycle. The Calvin cycle is ultimately going to produce half of a sugar molecule. The first part of photosynthesis, the light-dependent reactions, occur within all of the thylakoids of a chloroplast. In this image, we are looking at one thylakoid membrane. Inside is the thylakoid lumen. Outside is the fluid, the stroma of the chloroplast. Embedded within the phospholipid membrane of this thylakoid, we have several proteins. We have photosystem 2 on the left and photosystem 1 on the right. We abbreviate these as PS2 and PS1. They are named based off of when they were discovered. Between them, we have several proteins that constitute the electron transport chain, like we saw in the mitochondria. We have ferredoxin and NADP plus reductase, which is responsible for reducing NADP plus. And finally, we have ATP synthase, which we were introduced to in Chapter 9 in cellular respiration. Okay, so let's get into it. To begin, we're going to start by focusing on photosystem 2 and photosystem 1. Both of these are complexes of proteins. That means that there are many proteins that work together to form this structure. Around the outside, we have the light harvesting centers. Embedded within the light harvesting centers are all of the pigment molecules that absorb light energy. This could be chlorophyll A, chlorophyll B, carotenoids, xanthophylls, pheophytins, and more. We have many different pigment molecules. Most of our plants are going to have a variety of pigment molecules in the light harvesting centers of the photosystems. We saw this in Lab 7 when we used the TLC plate to separate out the different pigment molecules from spinach. Now there's a benefit to having a lot of different types of pigments, and that is that each one of them will absorb different wavelengths of energy. Remember that light is just packets of energy called photons, and they travel through space like a wave. The longer the wavelength, the lower the energy. And we usually absorb energy in the visible spectrum. When these pigment molecules are struck by one of these packets of energy, it's going to excite their electrons. And when their electrons get excited, they're actually going to jump to the next energy level. When an atom in its excited state is near another atom, it can actually transfer that energy to the other atom. Inside the light harvesting complex of our photosystems, we have two very special chlorophyll A molecules which are going to absorb at very specific wavelengths. In photosystem 2, they absorb at 680 nanometers, so we call this complex of chlorophyll A molecules P680. And in photosystem 1, they absorb best at 700 nanometers, and so we call that complex P700. And then finally, we have another molecule at the top of our complex, which is going to be the electron acceptor. When a photon strikes the electrons of a molecule and gets it into an excited state, that will then excite the other molecules around it until eventually that special complex of chlorophyll A are also excited. But they get so excited that their electrons don't just go to the next energy level, they actually leave the molecule and are accepted by the electron acceptor. At this point, since we've lost electrons, we would call this molecule P680 plus. In order to replenish the electrons that were lost by P680, we're going to break down a water molecule into an oxygen atom, two hydrogen ions, and the electrons that were with the hydrogens. These electrons can then be transported to P680 plus, restoring it back to P680. Meanwhile, those oxygen atoms can combine with other oxygen atoms to form oxygen gas. The electron acceptor of the photosystems is going to transfer the electron to the electron transport chain. Just like in cellular respiration, when the electron is passed from protein to protein in the chain, it releases a little bit of energy that can be used to do work. Just like in cellular respiration, that work is going to be the transport of hydrogen ions from a lower concentration to a higher concentration. So in this particular example, we are going from the stroma to the thylakoid lumen. This transport of hydrogen ions into the thylakoid lumen is going to create and maintain a high concentration inside of the thylakoid. We can allow these hydrogen ions to diffuse through a facilitated diffusion back into the stroma using ATP synthase. Just like in cellular respiration, that will cause ATP synthase to rotate, which will provide us the kinetic energy to combine ADP and phosphate, which would be in solution in the stroma, into ATP molecules. Meanwhile, photosystem 1 is also being struck by photons. These photons are going to excite the pigment molecules, which will then transfer that energy from one pigment molecule to the next until eventually P700 is excited. P700 gets so excited that it also jettisons its electrons, which are then picked up by the electron acceptor, and now we have P700 plus. The electrons lost by P700 are replenished by the electrons from the electron transport chain, which brings us back to P700. The electrons in the primary electron acceptor of photosystem 1 are then going to be transported to ferredoxin and then NADP plus reductase. NADP plus reductase is responsible for reducing or giving electrons to NADP plus, which is found in the stroma of our chloroplasts. This catalyzes the reaction of NADP plus and hydrogen ions into NADPH, which adopt the electrons and serve as a taxi for hydrogen and electrons. Once we've generated the ATP and the NADPH from the light-dependent reactions, we can use them to drive the Calvin cycle in the stroma of the chloroplasts. Now that we are halfway through, you should know the general equation for photosynthesis, be able to describe the structures of a leaf and the chloroplast, identify where the two steps to photosynthesis occur, and explain in detail what is happening in the light-dependent reactions, including the role of photons, chlorophyll, water, electrons, electron transport chain, ATP synthase, NADP plus reductase, NADPH, and the photosystems. You should also be able to explain the importance of maintaining an electrochemical gradient between the thylakoid lumen and the stroma.